Hello guys, we are yet once back again and we want to uh, have a lecture on electrophoresis. This is our second lecture on biophysics. As earlier established, these are my email addresses and telephone numbers. So let's begin by watching a video. But before that, let's see the course content. We have electrophoresis, that's background, technique, theory, equipment, and applications. Okay, so we'll begin this lecture by watching a short video.
Gel electrophoresis is the standard laboratory procedure for separating DNA by size for visualization and purification. Electrophoresis uses an electrical field to move the negatively charged DNA through an agarose gel matrix toward a positive electrode. Shorter DNA fragments, called bands, migrate through the gel more quickly than longer ones. You can determine the approximate length of a DNA fragment by running it on an agarose gel alongside a DNA ladder, which is a collection of DNA fragments of known lengths. In this video, we'll show you how to prepare an agarose gel using powdered agarose and buffer. Then we'll show you how to load your samples into the gel and perform electrophoresis to successfully separate your DNA bands. We'll also give you some tips and tricks to get great-looking gels every time. The first step in preparing an agarose gel is deciding what percentage of agarose to use. Agarose gels are commonly prepared in concentrations between 0.7% and 2%, depending on the size of the bands that need to be separated. The percentage of agarose in the gel solution determines the size of pores present in the gel matrix, which determines how quickly the DNA migrates down the length of the gel. To resolve smaller bands from each other, increase the agarose concentration to reduce the size of the pores the DNA is traveling through. To resolve larger bands, lower the percentage of agarose to increase the size of the pores. We will be preparing a 1% gel, which means we'll need to measure 1.5 grams of agarose and add it to 150 milliliters of 1x tris acetate EDTA buffer, called TAE buffer. You can also use another buffer, tris borate EDTA, also known as TBE buffer. TBE buffer is less prone to overheating than TAE and can be used for longer electrophoresis runs, which are often necessary to separate smaller bands of DNA. When using TBE to prepare the gel, be sure to fill the gel box with TBE too. Always fill the gel box with the same buffer that you use to prepare the gel. To melt the agarose, microwave the solution for 1 to 3 minutes until it is completely dissolved. Try not to overboil the solution, as some buffer will evaporate and will alter the final percentage of the agarose in the gel. Microwave the gel in pulses, swirling the flask occasionally as the solution heats up. Be careful when handling the flask, as the solution will be hot. Also, be careful when swirling the flask, as eruptive boiling can occur. After microwaving the agarose solution, wait until it cools down to 55 degrees Celsius. If the flask is cool enough for you to touch with just your gloved hand, then it's ready to pour. To reduce time, you can add ethidium bromide directly to the gel solution with a final concentration between 0.2 and 0.5 micrograms per milliliter. We're adding 5 microliters of our lab stock to 150 milliliters of agarose gel. Remember, ethidium bromide is a known mutagen. Wear a lab coat, eye protection, and gloves when working with this chemical. Once the gel has cooled, place a comb corresponding to the number of needed wells into the gel tray and slowly pour the gel. Make sure the tray is properly sealed around the outside so the gel doesn't pour out. Use a pipette tip to push away any bubbles in the solution. Let the newly poured gel sit at room temperature for up to 1 hour until completely solid, and then fill the gel electrophoresis chamber with the same buffer you used to prepare the gel. Pour until the gel is covered and make sure that the buffer enters the wells. Remember to load the DNA ladder next to your samples and write down what sample went into what well using the ladder as a reference. If you don't, your results may be very difficult to interpret. After the samples are loaded into the wells of the gel, connect the power source to the gel box and turn on the power. Remember, black is negative and red is positive. Always run to red. Most gels are run between 80 to 150 volts. Running at a higher voltage will make the bands migrate faster, but can also cause the gel to melt if the voltage is too high. Keep running the gel until the visible dye is 75% of the way down, which indicates you have good separation of your bands. This usually takes between 1 to 1.5 hours, depending on the size of your gel and the percentage of agarose that you used. Once your gel has finished running, turn off the power and disconnect the electrodes from the power source. Visualize your bands using ultraviolet light. Be careful when handling the gel, as it is fragile and can easily break. There are a few ways to increase the resolution or crispness of your DNA bands. You can use a gel comb that has wider or thinner teeth. You can run the gel for a longer period of time at lower voltage, or you can load less DNA into the well. We usually load between 100 to 150 nanograms of DNA in each well and run the gel at 100 volts for 1.5 hours to get the best looking bands for most of our samples. Gel electrophoresis is one of the fastest and least expensive ways to confirm the size of your DNA product. By using our tips and tricks, you will be able to get a great looking gel with clear, crisp bands to help you verify your samples every time. Thank you for watching Adgene's electrophoresis protocol video.
So, in 1807, Ferdinand Frederick Rose observed that application of a constant In 1807, Ferdinand Frederick Rose observed that application of a constant electric field caused clay particles dispersed in water to migrate, and he termed this process electrokinetic process. This was due to the presence of a charged interface between the particle surface and the surrounding fluid, currently applicable as an analytical technique in chemistry for the study of charged molecules and separation of macromolecules by size, charge, or binding affinity, and this process is termed electrophoresis. Electrophoresis is therefore the motion of dispersed particles in a fluid under the influence of spatially uniform electric field. In its application, charged molecules, mostly proteins and nucleic acids, which are negatively charged in solution, migrate in response to an electric field. Electrophoresis of a positively charged particles, and we know that positively charged particles are called cations. This process of moving positively charged particles through an electrophoretic setup is called cataphoresis. Electrophoresis of negatively charged particles, and these negative charged particles are anions, is called anaphoresis. So let's look at the rate of molecule migration and the factors that influence it. First and foremost, before molecules will migrate through a gel medium or through an electrophoretic setup, the field strength is a factor. Yes, we can see this is the electric field. This is the electric field. So, and these are the molecules or the particles. We have the negative particles, and one positive particles. No, these are all the electric field. These are the electric field. This is also electric field. It's all part. This is the particle which is positively charged. So it will move towards the positive. It will move towards the negative cathode. So it will be dragged either here or here. So this is the particle. It's moving. So the electric field is the field around it, and if the stronger it is, the faster the particles are moved through the gel matrix. Then we have the net charge. The net charge on the particle. If the particle is negatively charged, it will be drawn the particle is negatively charged, it will move to what? The positive anode. And if it is positively charged, it will move to the negative cathode. The size of the molecules is very important, because when the size of the molecule is big, it will find difficult to pass through the molecular sieve or the gel, because the gel it has pores inside that molecules traverse or move through. It's like when you are preparing zone, you sieve the flour. When you prepare zone, you sieve the flour. The reason why you sieve the flour is to let the fine particles or smaller particles to be sieved along with what you want to drink. Then the bigger ones become the waste, so you throw them away. So that is the gel, or that's the molecular sieve. So the shape of the molecules also is very important, because if they are rough shaped and they are not smooth, they find difficult to pass through the gel. The ionic strength of the molecules, the cations or the anions on it, will influence the electrostatic or the electricity or the charges. So if the ion is more positively charged, or if there is no charge on it, these all influence the movement. Viscosity of the medium, yes, is the buffer system that we are using to move the particles through the gel. Is the buffer system, is the buffer too thick, or is the buffer too light? If it is thick, if it is highly viscous, particles will find it difficult to move through the gel matrix. If the buffer is light, it will move particles faster. Temperature of the medium, yes, at high temperature, we have we have different types of buffers. We have phosphate buffer, then we also have trace buffer, we also have trace boric buffer, then we have trace acetate EDTA buffer. There are about 4: trace boric buffer, trace acetate EDTA buffer, we have phosphate buffer, then we have trace buffer itself. So this buffer system, if at a higher temperature, when something is heated up, it influences the movement of particles within it, and it has a lower temperature to influence the particles, because we assume that at a higher temperature there will be higher motion or movement of the particles. At a lower temperature, particles will find it difficult to move because they will be at a stationary stage. So this is so we therefore say that electrophoresis is usually done with gels formed in tubes or slabs or on a flat bed. So this is an example of an electrophoresis setup. This is the gel. This is what sample loading pipette. We have the cathode, that's negative charge, then the positive charge anode, the electrodes. We have one well here, another one buffer tray here, another buffer tray here. We put the buffer inside. You can see the blue color is the buffer, the sea blue color is the buffer. This is where we load the sample 1, 2, 3, 4, and this is the gel. The gel is supposed to be spelled GEL, gel. It is now hard. It was like a powder. You mix it with water, as you saw in the video. You mix it with water, then you blend it. You prepare like porridge. Then when the temperature is low, it becomes what? Hard. It's like TZ. When the temperature is high, it's a porridge, but when you cool it, it becomes solidified. So this is where the particles will be moving across using the buffer. The buffer will float. The buffer will flow over the particles. So the buffer will move. It will flow over the particles and send the particles to the gel. There are pores inside this gel. So the buffer will move this sample through the gel and separate its content into bands based on the molecular weight of the constituents of this sample. The same applies to this sample here. Same applies to the sample. Same applies to the sample here. Let's go look at the next slide. Okay, so in most units, the gel is mounted between two buffer chambers containing separate electrodes, so that the only electrical connection between the two chambers is through the gel. So the electrical connection, negative and positive, negative cathode, positive anode. So there is charges established within this system. So the movement, the only thing separating this electricity is what? The gel. So you can also see over here, this is the negative electrode, positive electrode. These are the separated, this is the sample 1, 2, 3, 4, 5. So the sample, this is a well, so this is the sample well, and this is the sample that has been separated. So this is the gel itself. So you can see this one will separate. So it means that this sample has 6 components. 1, 2, 3, 4, 5, 6. This sample has 2 components. 1, 2. This sample has 3 components. 1, 2, 3. This one has 4 components. This one has 6 components. And each of the components migrates according to what? Size, molecular weight, shape, and electrical charge. So you can see that those samples, the component of sample 1, this sample, moves, the components are 6. So the lighter ones will move faster than the heavy ones. So we can say that this one is very heavy, so it didn't move faster. This one is the lightest, so it moved faster. It moved to the gel faster. It is the smallest and the lightest, with a good shape and high and electrical charge over it. So this one has a negative electric charge because it's moving towards the positive electrode. So, but it has a small size. It is lighter, small size. It doesn't have much weight than this and this and this and this and this. The same applies to this one. This one, sample well. This sample, yellow sample inside, has 2 components. This one and this one. We can see that this one is lighter and smaller, and it has the electrical charge on it is negative, and the electrical charge is higher than this one. So it moved towards the positive electrode. This is the electrode. It moved towards the positive electrode. So it is lighter. You know, when you are running with a heavy, with somebody who is big, especially a woman who are so fat, very fat. You know, a slim woman will run faster than a fat woman. So this one is the slim woman. It will run faster than the fat woman. So that is how it is.